But remember that when you come to the Monday lesson, it often is either talking about the lab or is talking about a science skill that you will need. So last Friday, for example, I spoke a lot about bar graphs because we didn't have any labs for the week. Hi, Spencer. Thanks for joining us. We didn't have any labs last week, but we did. I did do a lesson on how to create bar graphs and how to work through bar graphs because you are going to have to do one for the lab that's coming up on Friday. So if you would like some assistance on that, their video is posted on the announcements. So you can watch that video, and the handout that goes with that lesson is posted in the description of the YouTube video. So if you open up the video that's embedded in the announcements, and then you open that up as a URL in a different web browser, then the worksheet that goes along with it will come up with it, okay? But we've got this lab 5.18 and 5.19 coming up next Friday. And that's going to mainly be focusing on seed germination and acidity. So we've been talking about ecosystems. We've been talking about abiotic factors and biotic factors and how when they work together, it impacts their life. And so what we're going to do with this experiment on Friday with this lab is we're going to see how something that's abiotic, remember abiotic means non-living, does interact with something that is living, that is biotic. So we're going to talk about a couple different things. I wanted to start with a quote. Again, I don't know if you know this, the last couple of times I've had lessons, I've talked really passionately about ecology in general, because I just think that it's so important for us to understand that our actions have a direct effect on our own ecosystem and our environment as a whole. So thinking about soil and dirt and everything, it's very easy to think to yourself, it's dirt, who cares, right? No, Christian, there's an assignment every day. If you take a look at the plan section, it will list it all what you need to do. So essentially all life depends upon the soil. There can be no life without soil and no soil without life. They have evolved together. And that quote is by an ecologist named Charles Kellogg, and he has been really involved with ecology and really spending some time heading some departments specifically about agriculture at the national level. And what he's saying is absolutely true. When we were looking at our lesson last week, we were looking at the food web, and one of the things we noticed was that bacteria was a very big part of that. And that's because some certain bacteria help with decomposing. And as we know, the decomposition of something puts nutrients back in the soil. And so we need to be really, really mindful about where we are, what we're doing with the soil. And so this lab is going to show you a direct relationship between abiotic factor and biotic factor. So we're going to talk about the two different parts of the lab. So 5.18 is lab part 1. And then lab part 2 is 5.19. And so we're going to go ahead and talk these through. And then at the very end of this, there is a discussion post. We're going to talk about what to answer in that discussion post and how to earn full credit. So first of all, we're going to be changing the acidity of the environment of seeds germinating. And what we mean by seed germination is we mean when it's fertilized and it starts to, like, the seed starts to crack and have, you know, those little first little signs of life, those first little growths that are the roots, right? So what do you think is going to happen? What do you think is going to happen if, thank you, the stems. What do you think is going to happen if we were to change the acidity levels, right? Do you think that more acid is going to be helpful or less acid is going to be helpful? What do you think is going to happen? It's going to be less helpful. Good. Okay, Christian. Good. Logan, Spencer, what are your thoughts? Less. Okay. Yeah. So it may not be as helpful, right? Which is crazy to think something that's not even living can prevent something else from being able to live well, right? So we're going to go ahead and talk about what the first part of this lab is. So there's two parts, remember, they are both assigned for Friday. So you have two lessons for Friday, and that is each part of this lab. So we're going to talk about the effects of acidity on seed germination 1, right? So that's the first part of the lab. So here are the steps for the both labs. So for both labs, overall, the entire thing on Friday is going to be the first thing is you're going to use an image analyzer tool to make observations of seeds. So you're going to be taking a look at several images, and I'll talk to you about that in a second. You're going to look at images and then make your observations and make a determination. Then the second part of your lab is going to be now you're going to be in charge of designing a solution. So now you need to come up with something that's going to help this plant grow. What is going to happen if you've got a situation where you have too much acidity? How are you going to fix that, right? How are you going to change that? And then the third, evaluate the solution. You're going to take a look at the design that you created and then think about it critically. Is this something that's going to help? And then you're going to think it through, test some of the things, and then you're going to refine the solution, which means you're going to go back and make any changes. And that's going to be your entire lab. Those two different parts, that's what this makes up this one big lab. Are there any questions on that so far? Okay. Good. Christian, you're already coming up with your hypothesis. I love it. The plant could grow quicker or it will rot faster. Interesting. I'm very excited. So, in the very first lab, when you get to lab 5.18, it is going to be based on the internet. So, it's going to be in those slides, it's embedded in those slides. And it's really easy to click through it. It's really easy to just keep going to the next slide. And then the next thing you know, you're going to be done with that lesson and on to the second one. And you're going to be like, what just happened? Where did it go? How did I miss it? So I kind of have two images to help you. So in one of the slides, you're going to be taking a look at several different seeds. You're going to be taking a look at 5 seeds, meaning 5 different types of species. And you're going to be looking at these images, and you're going to use this analyzer tool. So if you can see that these seeds have been laid out, and what you're trying to do is you're going to want to measure the root length for each of the 5 seeds on each day of the experiment. So what that means, you have 5 different seeds. There are 4 days of images, worth of images for each one. So you are going to need to create a data table, and you will need to create a way to measure it. So when we're taking a look at this first image, I do have it specifically pointing to one area, which I'll get to in a moment. But good question, Christian. You are supposed to measure the root. The root. So if we take a look, here is one of our seeds. And right here, we can see a little root is growing out of it. So what you're going to do, and when you get into the lesson and when you get into the slides, you'll notice that you'll be able to manipulate this a little better. So you'll be able to click down and move it. And then these numbers are embedded rulers. So what you might want to do is click one of these and then put it up against one of the rulers and try to measure it as best you can. And then you can use the zoom in to get a little closer so that you can be a little bit more specific. Now, like I was saying, it's really easy to click through these images. So I want to pause and show you that for each slide, it's a different slide for a different species of seeds. And how you get to the different days is this little album picture. So this little album picture is going to, once you click on that, it drops down here. So this day 1, day 2, day 3, day 4 image, That's how you're going to go from day to day to see the different lengths of the root for that individual species. Okay. So really important because it's so easy to miss. I mean, when I was getting this lesson together and going through it myself, I noticed how easy it is to skip over it. So really just make sure that you're taking your time, that you click and explore with these tools to see how you can best use them to measure the root growth accurately. And then pay attention to any of the labels because some of them are going to be with vinegar and some of them are going to be without vinegar. So you really want, and the vinegar is what we're using to increase the acidity. So you really want to be mindful, okay? You really want to be paying attention to the labels, all of the different buttons, the different things that you can use, the tools, and then utilize that to create your observations, your measurements. Remember to include qualitative, quality, senses, right? And quantitative measurements, numbers. So that's going to be very much in the way of the length. So that's going to be the first part. Are there any questions in part one? I've got another slide to go over too. I think is still part of it. Okay. Any questions about the analyzer tool, the image analyzer? So Christian, you're actually not going to be, this is a virtual lab. So the images it's already been done for you. So you're going to be looking at the seeds, but yes, what the experimenter has done is oftentimes when people germinate seeds, they put it in a single layer of paper towels. And you can do this too with any seeds. You can do this as an extra experiment where you take a couple of seeds, you take a paper towel, you want to wet the paper towel, open it back up, put the seeds in the paper towel, put it over another wet paper towel, and then you want to leave it somewhere where it can then utilize that water and start to sprout. And so what this experimenter did is they did one with the environment of just water and then one where they included water and acid, the vinegar, into that paper towel and how that affected the plant's ability to germinate and sprout and grow roots and stems, right? So that's what they did. And they kind of explain that a little bit more when you get into the lab. So if we go into the next slide, the second part of part one. So the first part is taking a look at the images. And then the second thing you're going to want to do, and you have to pay close attention because this is written in the slides, but is something that can be easily overlooked. So make sure that when you're clicking it through slide, you're really paying attention to the instructions because they ask you to compare your findings. So then after you've done, after you've looked at the analyzer tool, you've looked at the images, the next thing they ask you to do is to conduct a search on the effects of acidity on seed germination. So this is something that you can do by plugging it into Google. You can search it, you can do this and figure and use it as a way to figure out what other research has been done. What have the other findings? And please do not do this until after you've made your own analysis, right? Because I know it's really tempting. However, you really miss out on the skills. You really miss out on your own practice in terms of making observations and then using your observations to make a conclusion. Those are really big skills that if you were to skip that and just go straight to researching what happened, you're missing out on a lot of the learning. So even though it seems like it's an easier, faster way, it's really a disservice because you're going to be losing out on opportunities to grow and strengthen your understanding, right? And then once you find the research on the effects of acidity and seed germination, then you're going to compare the findings to your experiment. So then you get to look, okay, what has been done and what did I find? And see, okay, were you in line with the results from your search, or was there maybe a level of misunderstanding, right? Was it you or was it them? So that's going to be the first part. So that was lab 5.18. So that was the first part of the lab. Once you're done with that, you move right into lab 5.19. And that's titled The Effects of Acidity in Seed Germination 2. And this is when you are going to design, evaluate, and refine a solution. So thinking about this, right? We know that we are right now in the throes of climate change, and that climate change is really starting to affect the way that things grow, the weather patterns, and so forth. Part of that climate change is the human activity. There is heavy evidence that tells us that because of human activity, we have more pollution, more methane gas, more carbon emissions into the air and into the atmosphere. And when it starts to rain, that acidity, or that all of those elements are built up, and then they fall down with the rain. So we start to see something called acid rain. And, one or two times that it rains with a little bit higher of an acid concentration isn't going to change much. But as we know, living in Oregon, where it rains for a long period of time, this is the thing. If you were to get rain on you while you're walking through, you're going to be fine, right? You're not going to melt, you're not going to burn or anything like that. But as the water accumulates into the soil, and that water is carrying those carbon emissions and all of these extra pollutants that are in the air with it, it starts to increase and it starts to add up. So now what we're starting to do is, even though in a laboratory setting, we're seeing the effects of acidity on seed germination, we want to use that example and understanding and apply it to a possible solution to something that we can do here in the ecosystem around us. So the lab for this second part is going to be, the objective. So the point of it is for you at the very end of this to be able to say, I can conduct an investigation that shows how an environmental factor can affect, and that should say a life form. That was my misspelled. Can affect a life form. So we're starting to see, even though we're doing like mini experiments in the lab, how we can apply this to the real life. So this is the materials that were used. We've got vinegar, resealable plastic, seeds, baking soda or antacid tablets. You'll need a metric ruler and several paper towels. So this is when you're going to be collecting these. Now, there are things that are coming from our experiment boxes, so our lab kits. That should be the seeds, metric ruler. But at home, if you don't have vinegar, and it can be any type of vinegar, it could be white vinegar, apple cider vinegar. There's a variety of different kinds, red wine vinegar, doesn't matter, any type of vinegar that's pretty much found in the kitchen. And it's typically a household baking or cooking item. But if you don't have any of these materials, please let me know. I will help figure it out, whether that's I order it on Instacart and get it delivered to you, whether that's me bringing it to myself or bring it to you myself, whatever that looks like. If you don't have any of these objects or these items, let me know and I can help you guys out, okay? But these are the materials that you're going to need to have. And this is already settled and written in your lab as well. So the question that we're really going to be asking is, how can we minimize the impact of acid rain on seed germination? And so when we talk about minimizing the impact of acid, we have to understand a little bit about acid and bases. Now, we usually think of the pH scale, which we've talked about in biology throughout the year. The pH scale is how we determine how acidic something is, how reactive it is, and how basic it is. Now, when we talk about basics and acidics, we're really talking about their level of interactions with elements, okay, with different elements. So things like battery acids. So batteries are actually a chemical reaction. So when you think of a battery, it's really important to make sure that it doesn't leak, right? Because if you do, it's really, really acidic. But on the other hand, we also have something like bleach. That's really, really basic. Both of these things are not good when you ingest it. So we really want to be careful. But when we mix something, when we mix an acid with a base, they tend to cancel each other out. So when you're answering this question, take a look at this pH scale that they include in the slides, because this is going to help you determine what exactly, Christian, it can cause burns. And this is going to help you determine how you would go about designing your solution. Right? So there's a little thing here. If you mix something acidic with something basic, you can get a neutral mixture. So for example, we can mix vinegar with baking soda, and that's going to turn that solution into a neutral pH. Now, we often think of baking soda and vinegar as, if you've ever seen those like paper mache volcanoes, right? Like the classic science project that you see, and you have it bubbling out, that is the reaction of vinegar and baking soda. So there is this real life interaction that you can witness and see. So you're going to start to think about, using the information that you know, what can I do that's going to neutralize the acid in the environment? So when you're doing this, remember, this is something that you get to design. So while you're designing this solution to the acid rain on seed germination, you want to take into consideration two things. You're really only going to want to conduct tests with the household items, because that is the safest. Okay, we're not asking if you have a parent that has access to higher concentrated chemicals. That is also a safety concern. So we really want to keep it to those materials that I listed on the page before, and it's already listed in your lab as well. And the second thing is, what controls are you using to test your design solution? So remember, we only want to be changing one variable at a time. So what's going to stay the same for everything else? What temperature, what amount, where, what kind of seeds, right? All of these things need to remain the same throughout all of your experiments. And that's not all, right? That's just the starting point. So really making sure that you're only changing one thing at a time is going to be huge. Now remember, you are going to be creating a solution. You are creating your experiment. That being said, there is a general procedure that you can use that you should not be only sticking to. This should be the starting point, and then you should be changing the procedure for your specific solution. So for example, you are only going to use one type of seed. You're going to want to use multiple seeds for each test conducted. You're going to soak a paper towel in each liquid to be tested, and then place the seeds in a paper towel. Kind of what I was saying, right? You're going to soak the paper towel, you're going to open it up, and then you're going to layer it on the bottom and in the top of your seeds. You may want to mix your ingredients. So for example, we're trying to solve for a solution, right? We're trying to come up with a way that we can neutralize the acid to allow for seed germination. And that means that you may want to mix a couple of create your solutions, right? And mix it together before you soak the paper towel in it, okay? And then you're going to place the seeds and paper towel in a sealable baggie, and you're going to monitor the seeds over the course of several days. Now, this works out really nicely because the lab is on Friday. So you should be at the latest conducting this on Friday, and then allow the seeds to go over the weekend, checking on it once a day, at most twice a day. And then once you check it once or twice a day, then there is a discussion post that's based on what your findings have been. But remember, these are the general procedures. So when you're writing your procedure, when you're figuring out your procedure, you really want to be specific. Now I'm going to go back to this image. These are a couple of examples. These are pretty household items: soap, bleach, some sort of ammonia, baking soda. You're not going to test blood. Blood is off limits. Absolutely no blood. None whatsoever. Okay? None of that. But you can start to look, okay, milk is a little basic, right? When we think about milk, or excuse me, it's neutral, but it's a little bit more acidic. We often think of milk as a way to help with acid and digestion. When we're starting, our stomach acid is so active that it starts to go up our esophagus and it starts to hurt. And we can drink milk to try to neutralize it. So you can take a look at some ideas. You can do a little bit of research, make that mixture, that liquid mixture, before you dip your paper towel into it. And you're going to want to write about what you did and how much, right? Don't just eyeball it. You're going to want to measure this out. So whether that's using a scale, whether that came with the box, whether that's measuring it in the graduated cylinder, whatever it is, you're going to want to measure your solution. Then you're going to go ahead, do the experiment, make your observations very similar to what we did with the image analyzer tool, except now this time it's going to be in your face. So you're going to use that metric ruler to measure the root and see what the effects are of your solution. And once you do the solution, then you can start to refine it. You're going to evaluate it. How effective was it? And then refine it. What would you have done differently? So that's going to be for Friday. After you've had a chance to conduct your experiment, then you can go on to your discussion post, which as of right now is not due until next week. So when you're doing your discussions, think about this, because a lot of people have been losing points for not writing their discussion post fully. And that is reading the discussion prompt carefully and then writing your response. Think about how your responses should be well written and clearly address the issue being discussed. So that means answering all of the questions, not just one, maybe not just two. If there are 3 or 4, you should be having 3 or 4 answers within your post. And I check it regularly. Excuse me. So making sure that your post is being appropriate, that you are being kind to one another and responding. Remember that you have to respond to 3 other posts. But here are the 4 questions for your discussion. You are going to describe your design solution and how you conducted your experiment. That's 1 of 4 of your discussion. Then, was your solution successful? Explain by citing evidence from your experiment. So you're going to use the data that you collected from your observations, and you're going to include that in your discussion post. Then how would you refine it? How would you make it better, right? What would you change for next time? And how does your design solution compare to the designs of other students? So that's when you are looking for your peers, and you can respond by talking about it and then including that in your own post. Okay, so that's kind of the general overview of your lab. And that sounds like a lot, and it is, but you're also getting to like do a real live test. So this is also a really exciting opportunity for you guys to see exactly something that we are challenging and battling with every single day. It is not uncommon to be dealing with acid changing in our soil and how that affects the plants that live here, right? So really thinking about that, because again, we are part of the ecosystem, right? So understanding that when something has an effect, when it affects everything else. So if there's acid rain falling into the soil, that's going to affect the life that grows there. And if that affects the plant life, that affects the animals and so forth. Okay. But that's what I have for you guys. Do any of you guys have any questions on the lab that's coming up on Friday? Great. Well, if you have no questions, that's everything. So you guys can, that's everything I have for you. We ended a little early, so bye. If you guys have somewhere else to go, I completely understand. Let me know if you have any questions on the lab.